A plant tissue culture laboratory, whether for research or for commercial purpose, should provide certain basic facilities, such as washing and storage of glassware, plasticware and other labwares preparation, sterilization and storage of nutrient media, aseptic manipulation of plant material, maintenance of cultures under controlled conditions of temperature, light, and humidity, observation of cultures and hardening of in vitro developed plants. The extent of sophistication in terms of equipment and facilities depends on the need and the funds available. Therefore, establishment of a new tissue culture facility requiring ingenuity and careful planning. Washing room. For a washroom, the following facilities are required: adequate supply of good quality running water and an acid and alkali-resistant big sink. Adequate steel or plastic buckets and tubs are required for soaking culture vials and other labwares used in medium preparation. Brushes of various sizes and shapes are essential for cleaning glassware. Even if good quality water is available, it cannot be used for final washing of labwares or for medium preparation as it contains impurities such as inorganic and organic compounds, dissolved gases, particulate debris, and microorganisms. Water could be purified through distillation, deionization, or reverse osmosis. Sometimes a combination of two or more is required. Water purity is measured in terms of resistivity, ohms chem, 1, or its reciprocal, II, conductivity, mos chem, 1. Water for tissue culture should ideally have a conductivity of 5.0 ohms chem, 1, although a conductivity level up to 15 ohms chem, 1 is acceptable. Media preparation room. The media room is provided with a working table in the center and benches along the wall, the tops of which are either covered with granite or laminated board. The tables and benches should be at a height suitable for working while standing, and the space below them could be fitted with drawers and cupboards for storage purposes. The benches are required for keeping balances, feet meter, magnetic stirrers, hot plates, etc. A top-loading electronic balance with tare for weighing large quantities and an analytical balance for small quantities of chemicals must be provided. The balances should be isolated in a small chamber if the media room also houses the autoclave. In a large commercial laboratory, it will be of help to have an automatic media dispenser. Glasswares, plasticwares. In a tissue culture laboratory culture, vessels are required in bulk. Depending on the type of work, adequate supplies of these should be maintained. For standard tissue culture work, rimless test tubes, 25x150m, are widely used. The culture tubes are important for culture initiation and establishment even in a commercial setup. For further mass multiplication, larger containers, such as jam bottles or other wide-mouth bottles, are required. Erlenmeyer flasks have also been used as culture vessels. Only borosilicate or Pyrex glassware should be used. Plastic culture vials, autoclavable and pre-sterilized, have replaced glass culture vials to a large extent. A wide range of pre-sterilized disposable culture vials made of clear plastic, especially designed for protoplast, cell, tissue, and organ culture, are available in the market under different brand names. The pre-sterilized plastic petri dishes, jars, screw cap bottles, and various cell culture plates come with their closures. For culture tubes and flasks, traditionally non-absorbent cotton plugs wrapped in a single layer of cheesecloth have been used as a closure. Cotton plugs provide excellent aeration, but the medium dehydrates very fast. On the other hand, polypropylene caps reduce the rate of medium desiccation, but increase moisture and gaseous accumulation within the container. However, it is important to ensure that the closure allows proper aseptic aeration and does not inhibit the growth of culture materials. Parafilm Nesco film, commonly used to seal petri dishes, releases butylated hydroxytoluene, which is toxic to the cultured plant material. Alternately, one can use cling film for sealing, as 2-ethylene. 1-hexanol released by it is not inhibitory to culture material. Culture vessels made of polypropylene transmit 65 light, and those made of polycarbonate transmit almost 100 light. Gas-permeable fluorocarbonate vessels are available for use with plant materials that are sensitive to gas buildup within the culture vials. Kozai, 1991. Besides culture vials, various other glass, AI plasticware such as beakers, measuring cylinders, pipettes, etc. of various sizes are required for media preparation. Transfer room. No special requirements for a transfer room except that a high degree of cleanliness and a worker-friendly environment is to be maintained. Transfer hoods are placed in this room to carry out aseptic manipulation. Often the culture medium to be used is also stored here. although, where possible, a separate storeroom within the clean areas is demarcated for this purpose. To transport culture medium or cultures in and out of the transfer room, trolleys with one or more shelves are helpful. These trolleys may also serve as side benches for the operator to hold extra culture medium, stock cultures, and your freshly raised cultures until transferred to the growth room. Since fire heat is constantly used in the transfer hoods, it is advisable to keep a fire extinguisher in this area. Growth room. The inoculated culture vials are transferred for incubation to a growth room with controlled temperature and light conditions. It is of paramount importance to maintain cleanliness in this area. It can be achieved by having positive air pressure in the clean areas or an overhead air curtain at the entry to remove surface dust. This room should have one door, and the window should be avoided to prevent external light from interfering with the internal light cycle. Desirably, the junction of walls should be rounded rather than angular to prevent cobwebs. The wall paint should be of semi or high gloss to withstand repeated cleaning. Growth rooms, being closed units, require devices to control temperature and light. Temperature is controlled by air conditioning. Adequate precautions should be taken to filter the cooled air to prevent dust, spores, etc. from reaching the clean area. The air conditioners and heaters are hooked to temperature controllers to maintain the temperature at 25°C. Generally, cool white fluorescent lamps are used for growth rooms because of the uniform light intensity they emit. Cultures are normally maintained in diffuse light, 3000–5000 lux. The low light intensity can be achieved in 3.5 feet wide shelves by installing 3 tubes 1 foot above the surface of the shelf. A sheet of aluminum foil or coat of aluminum paint provided above the tubes maximizes light intensity below. Some provision should also be made for growing cultures in total darkness or under high light intensities. Automatic timers are used to regulate the photo period. If the relative humidity in the growth room falls below 50, humidifiers are required to be used to prevent medium from drying rapidly. Dehumidifiers may be required when the humidity is very high, particularly during the rainy season, to prevent cotton plugs from becoming damp or condensing of water droplets, as both may increase chances of contamination of cultures. PA culture vials in the growth room are maintained on specially designed shelving unit that are either stationary or movable. Stationary shelves may be fixed on walls of the room, or could be fitted into angular iron frames to form culture racks that are placed conveniently in the room. Alternately, the racks could be on roller coaster wheels that allows efficient use of space. Open shelves are generally preferred because of better air circulation. Each shelf is provided with a separate set of fluorescent tubes. In case the cultures are to be maintained under different photo period and temperature regimes, it is advisable to have more than one growth room. Culture flasks, jars, and petri dishes can be placed directly on the shelves or in trays of suitable size, while culture tubes require some support such as a metallic rack, which can hold 20-24 tubes. In commercial companies, where large quantities of culture vials have to be moved, it is not only convenient, but also time-saving to use autoclavable plastic or epoxy-coated metallic trays for holding the culture vial. The culture vials and the trays holding them should be appropriately labeled to avoid mixing up. For transportation of culture vessels, cart trolleys may be used. The culture room should also have a shaking machine, either of the horizontal type or the rotary type, if cell suspension cultures are to be grown. Cold storage. In a commercial setup, it is necessary to have a cold storage maintained at 2 Farok for temperate plants and 15 Ok for tropical plants. These rooms are used to give treatment for breaking dormancy of some plant materials, storing of cultures to schedule workload, maintain mother cultures, and to hold harvested plants. Greenhouse. In order to grow the mother plants and to acclimatize in vitro produced plants, a tissue culture laboratory should have a greenhouse made of glass, polythene, or polycarbonate. This facility should have a provision to maintain high humidity. It would be desirable to have a potting room adjacent to this facility. A separate autoclave might be required in this area if one wants to sterilize the potting mixture.